Okay, good afternoon, everyone. My name is Xiao. I'm the head of platform R&D department in GenScript ProBio. Today is my great pleasure to share with you how GenScript can provide one-stop CMC solution, mRNA vaccine and therapeutics. I'll start by these are my contents, introduction to ProBio and our one-stop solution from plasmid to mRNA. This is our business footprint. Started in 2018, we completed the first plasmid process development facility in China area. And since then, I won't go into the details, but I want to highlight it. I want to highlight that since then we've helped Legend ProBio to obtain the first CAR-T IND approval, the first TCR-T project in China, the first mRNA IND approval in China, as well as the first CRISPR gene editing IND approval in China. And up to now, we've obtained more than 20 approved IND in China as well as in global. So this is a demonstration of our global partnerships and our track record. We've served 300 plus plasmid and viral vector projects, ranging our key accounts ranging from China to US, EU, as well as Asia Pacific. And these are three examples of disclosable partners that we've worked with. And so in very short time frame from 2020 to 22, their project pipelines were proceeding very fast, with some of them at the phase 3 and EUA in Asia as well. So we have constructed our facilities in such a way to provide consistent support for our customers from early research all the way to commercialization. And up to now, we have already we have our plasmid vector as well as mRNA process development and GMP facilities ready. We are also planning for further expansion of the facilities for each respective application field. And we have also a fully commercialized GMP center dedicated for gene and cell therapy CDMO business being planned in both China as well as the US region. So the facilities are so this is the landscape of our plasmid facility and mRNA facility, including the batch size and the production lines. Overall, the facilities are designed to avoid cross-contaminations, yet to be flexible enough to ensure the fast delivery of the products. We have a very strong project management team, which that is comprised of 100%. So all the project managers as an important interface between us and the customers. All of them have obtained PMP certificate. Larger than 60% of them they have global project management experience. Larger than 30% of them obtain PhD degrees. And for CDMO, our customer goes first, as well as our quality system. So we, in quality management system, we have a well-established quality system that is compliant with China, US, EU, WHO, these GMP systems. And we have a complete quality management organization to help us safeguard the life cycle management of the product quality. So this is an overview of our one-stop solution from plasmid to mRNA in each respective applications. Plasmid, linearized plasmid to IVT mRNA, and to mRNA encapsulated by lipid nanoparticles. And we have dedicated product offerings in the early preclinical development, IND filing package, and also early phase and late phase clinical trial material supply, all the way to fully commercialized GMP grade. So if you look at our integrated platform timeline, there we can take make the best use of the plasmid integrated mRNA service that can shorten our overall project cycle, turnaround time from 10 months for first GMP batch release to shorten it to 6 months from for this tox material release. And as you can see here, if you split the plasmid and mRNA, it will take up to 10 months. But if you put plasmid and mRNA under the same roof, which we here in ProBio seamlessly integrated the plasmid and the mRNA CMC. As soon as the plasmid sample is ready, we start mRNA CMC right away. And when the plasmid first GM Pro material is ready, we can start the tox batch production for mRNA right away. So it's really a very close integration of the plasmid and mRNA, which guarantees the very short timeline, the overall CMC product development. In the next few slides, I will give you a little bit, let's zoom in a little bit to the mRNA. I'll talk about the challenges and the strategies that we use in ProBio. So to borrow a slide from Moderna on the mRNA application, the layout of application field is very broad, including preventative vaccines, cancer vaccines, tumor immune-related therapies, regenerative, autoimmune, and the circulatory-related therapeutics. Also, there's inhaled therapy, systemic intracellular therapy, so on and so forth. And in addition to these well-known mRNA vaccines, mRNA can also be used as protein replacement therapies for instance, antibodies and fusion proteins. And also there's CRISPR-mediated gene editing therapy are also harnessing the mRNA in doing harnessing the mRNA strength. So if we look at the CMC challenges, it really in each respective application of mRNA therapeutics mentioned before, their stability, efficiency, immunogenicity, safety, and effectiveness are the key points to consider during the CMC development. mRNA sequence design, process development, and design space exploration, as well as mRNA delivery, are the key impacting factors to address these CMC challenges. These are also what our mRNA platform has been focusing on. And how to address these challenges during the CMC development? The quality by design concept is a powerful tool that is rooted in our scientists' daily work. It really stresses the gradual understanding of the molecule and process, leading to the implementation of appropriate quality control strategy based on the risk assessment during the CMC development. So starting from the molecule itself, it will have a huge impact on the process as well as the control parameters. And as a CDMO, we do process design for our customers in a broader range, including raw material strategy, capacity and scale design, process design itself, as well as the economical solutions to make sure that the process that we develop and design is economically feasible for future commercialization. Control strategies and checkpoints, we have during the CMC for process control and the risk mediation is very important. There we have strong analytics and the quality control group that can help us to control the quality of the products. And of course, as a CDMO, we want this quality by design wheel to roll, leading to accumulation of process know-how and database that we develop to capture this knowledge. And with that, we can make sure that at each incoming new project, we always find more appropriate and more suitable CMC strategy to proceed the project. So in mRNA manufacturing process, we have established 4 technical platforms, starting from circular plasmid to linearized plasmid to in vitro transcription of mRNA with 2 different capping methods, which are enzymatic capping, the traditional one, and the co-transcriptional capping. And there we are partnered with TriLink to provide this clean cap technology. And we also have LNP formulation platform to help us until the DP of the mRNA. And further on, I want to zoom into more details of the mRNA IVT process platform, as well as our formulation process platform. So our IVT platform is well established. It can offer multiple capping methods, as I mentioned before, with the yield at 10 gram per liter level. So this page shows the process of enzymatic capping, which is an enzyme such as we use vaccinia capping enzyme as a typical enzyme to convert the 5' phosphate group to the cap 0 structure. The cap 0 can then be modified to the cap 1 structure by methyltransferase. And we have tested different suppliers indicated here, including domestic as well as international enzyme vendors. And the overall capping efficiency is comparable, and it's very high. It's larger than 95%. We have done a lot of exploration during the process design. We explore the process design space of enzymatic capping method using design of experiments to explore a wide range of process conditions, and then increase the granularity to find the key rate limiting steps, and finally to block the process parameters and the input parameters for each process steps. With respect to each substrate addition, for instance, the substrate addition time and rate, and so on and so forth. Amongst them, we also pay full attention to the screening of IVT raw materials. As you may know, that the IVT for mRNA manufacturing, the raw material is quite critical and it's very costly as well. So there we consider the supplier qualifications, the quality of the product, effectiveness, and the raw material cost and the accessibility very early on in the initiation of the projects. So scale up is another key factor to developing a robust process. And during early process development, we already pay attention to for instance, reaction time, temperature, and flow dynamics to zoom into these mass transfer related factors that can guarantee later on the success scale up prediction of the processes. So as a result of the DOE and the scale up studies, we achieved quite a very excellent process platform. So here we show in some real world cases. In this study, there are actually two projects, two molecules. These two molecules are known by the customers that are that were is very poor capping efficiency. So the mRNA molecules are very difficult to be capped due to its molecular inherent attributes. But after rounds of DOE experiments, we managed to increase the capping efficiency from the original 40% up to 90% and maintained very stable during our process lock-in batch as well as the further scale up batch. And now this project have completed 6 batches of GMP manufacturing for these two projects, and the process robustness and stability are both very good. And also as you can see here, the mRNA yield as well as the recovery rate. So the overall performance of the process for these two projects are very well. Apart from the enzymatic capping process, which I mentioned in the previous slides, we also partner with TriLink to provide the clean cap, a co-transcriptional capping process. With the co-transcriptional or one-step capping methods, the mRNA manufacturing process can be greatly simplified, resulting better scalability of the process, lower material cost, and therefore greater commercial feasibility. So here we have some results tested using TriLink CleanCap and the other and some cap analogs from the other vendors as a comparison here. The capping efficiency were excellent and reproducible, as well as for the mRNA yield, which are not impacted by different cap analogs that we use. All of them showed very high production yield. And when it comes to, so after the in vitro transcription of the mRNA, typically we have to do downstream process to remove the process and the product-related impurities. And we do this through, for instance, UFDF and affinity chromatography, further polish, and sterile filtration. As you can see, there are also a selection of polishing steps that we can use if further removal of the dsRNA, for instance, or truncated RNA is needed. And the overall recovery rate depends on the process that we develop, can range from 40% to 80%. So we talked about process and capping process. Apart from that, very strong quality or strong analytical platform is also very important, because the quality analysis is really the eyesight for our for process development. So in there, leveraging our strong capabilities in gene synthesis and the nucleic acid development, we have established a strong AD and QC platform, and there we keep pace with the latest regulatory guidelines, participated in drafting domestic guidelines in cell and gene therapy field. And our system is compliant with China, US, EU GMP system and passed audits without any critical findings. We have a state-of-the-art analytical methods, including the release testing as well as characterization. And all these methods that we use, the equipment are being computer system validated. And we do that all, we can also provide in-house capability for establishing these validation procedures. So this slide will give some demonstration for our quality analytics platform. So ranging from mRNA purity analytics, so we have orthogonal release method like CE, HPLC, or gel electrophoresis to look at the purity of mRNA from different angles. Also, we have accurate and high resolution LC-MOS system for capping characterization, so capping efficiency characterization, and as well as for the poly-A characterization. We have also in vitro and in vivo pharmacology group characterize the mRNA functionality, as well as later on the LNP encapsulated mRNA. So these testings were used during the process development, sometimes during for instance, also used to characterize using different purification methods or different vendor selection. We can really have it can really give us the confidence for the produced mRNA functionality right from the beginning during the process development or material selection phase. So we have also ongoing studies on the stability of mRNA, which is known as a critical issue for these mRNA molecules. So mRNA was known to be fragile and hard to store. The data that is being accumulated, and so far we obtained very good stability of the mRNA, various in use as well as storage conditions. So in the next section, I will give some introduction to our formulation process. So LNP development platform. We have built the LNP platform to be universally applied. So with different formulation that we tested our platform and the encapsulation methods and the scale-up strategy that we use were very that can apply widely to different LNP formulations and ionized lipids. So we have established this microfluidics based LNP process platform from process development up to GMP scale productions. The equipment has built-in functions to keep similar flow dynamics between different scales and similar mixing pattern for the LNP encapsulation, which ensures the consistent scale-up of the LNP process from the laboratory level to the GMP level. And we have tested the different LNP formulations, for instance, the Moderna and BioNTech formulation, as well as some our partners ionized lipids. So all of them showed quite good physiochemical attributes on our LNP platform. The size, morphology, and the physiochemical characteristics are very important for this LNP quality and the LNP functionality that determines the mRNA biodistribution as well as cellular uptake, therefore the efficacy of the mRNA. And these characteristics can be optimized through, for instance, formulation optimization, process optimization, like flow rate optimization, and mixing parameters. So the LNP platform, as I mentioned before, that we have is very universal for quite a wide range of formulations that we tested. The performance also across different applications are very satisfying and quite consistent. So in this slide, we demonstrate we want to show that the LNP process is very robust and scalable. So all the way from milligram level to hundreds of. Actually, we did the most recent data is already nearly a gram level LNP process, and so the scale up from the lab scale to GMP scale is demonstrate the scalability of the performance, as well as on for different mRNA molecules with different length. and also the batch-to-batch variation is very small during the scale-up. So for the encapsulated mRNA, apart from the physiochemical analytics, for instance, here we also have the cryogenic transmission electron microscopy. We use it to check the morphology of products, and we also have cell-based assay and in vivo methods to measure the mRNA activities. And again, these methods have been tested on various mRNA types, like, for example, linear RNA, self-amplifying RNA, circular RNA, and also on the DNA, tested the delivery using the LNP system that we have. We also can provide, can develop the lyophilization process for the mRNA drug products. So I think with that, so this is the sum up slides on our plasmid and mRNA platform. So in this presentation, we focus on mRNA, but on the other hand, our plasmid platform is also very strong and very experienced, which guaranteed to pave a very strong foundation for our next step mRNA development. So I want to iterate here that we can provide the one-stop solution from plasmid to linearized plasmid, IVT, and all the way to the mRNA LNP drug product development. And we have a competitive project cycle. Within 6 months, we can provide mRNA tox production, and the whole package management ensures the short turnaround time of the projects with a very strong project management team to align our customers' needs and our development progress. And with that, we and our customers are on the same page for the project development, and the risks are appropriately controlled throughout the process. So there are some technical advantages. I'm not going to details for the sake of time. And we also listed here that some our track records in mRNA development field, some collaborations that are disclosable. And with that, I want to thank you for your attention. I listed here 2 of our commercial BD managers, Jennifer and Junil, for you to contact further if you have any queries in future collaborations. Thank you very much.